Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Hello students, today we are studying chapter Gene, its nature, expression and regulation, and in that we are doing topic parts of an operon. Let's study how does an operon function and what are its different parts. Parts of lac operon or parts of operon. In 1926, a French microbiologist suggested that there was something called as lac operon or inducible operon present in E. coli. Now, how does that mean and what does it work for? In simple language, an operon is nothing but a switch on and switch off machinery that's present in E. coli. For example, if the E. coli needs to convert its glucose into some other form, that is what decided by the operon, or operon functions according to that. Whether it has to do certain process or it doesn't has to do certain process is what is decided by the operon. Today we are trying to see the various parts or various segment an operon would consist of. The three different regions that an operon actually consists of is the regulatory region, the promoter region and the operator region. Now if we talk about the regulatory region, students, this is the region where protein synthesis is wholly and solely decided, or this is the region which consists of genes that would help to make that protein. For example, galactosidase, especially in the case of E. coli, would be made only if the regulatory region is present. If any hindrance comes to the regulatory region, obviously a protein won't be made. Second one, the promoter region. Inside transcription also we have studied that the promoter region is ahead, and to this promoter region what binds is your RNA polymerase. Now first RNA polymerase attaches to the promoter region and does the synthesis of the transcription unit or mRNA. So promoter region is the starting base. The next one, the operator segment. Now when I talk about operator segment, if I've told you that operon is a switch on and switch off mechanism, the operator segment plays a very vital role, students. To the operator region, what happens is your repressor proteins, or you can say the proteins which control or stop the synthesis when the cell doesn't want it, and the region where repressor protein binds is this region, or you can say is the operator region. Parts of operon, or you can say parts of an lac operon or lactose operon. The first region is the regulatory region, or the region that actually regulates the protein synthesis. Now students, this regulatory region is the one that codes for the repressor protein molecule, and we all know that if the repressor protein molecule is absent, then the switch on and the switch off won't be available in the system of E. coli or any protein synthesis. We are studying about the regulatory gene. Now the repressor molecule binds to the operator region, or you can say the starting region, and prevent the RNA polymerase, the polymerase that actually makes the mRNA of an enzyme, to bind to the promoter segment. Next one is the promoter region. Now this is a short sequence where RNA polymerase or R polymerase first comes in attached. Now, if we talk about the promoter region, it initiates the transcription of structural genes and control the rate of mRNA, or you talk about messenger RNA. The next region is the operator region. Now, the operator region is nothing but a short sequence of DNA that's present where the repressor proteins binds. Now, this short sequence of lac operon where the repressor proteins binds prevent the RNA polymerase, or you can say the RNA synthesizing protein polymerase, from attaching to the promoter. And if students, RNA polymerase is not binding to the promoter, don't you all think so that proteins won't be synthesized? And if proteins won't be synthesized, obviously a function would be hampered or stopped. Now, this is the on or off switch of transcription which I was talking about. Students, all these three different regions, whether it is your promoter region, whether it is your operator region, or whether it was your other regions present on lac operon, all these regions work in co-association with each other so that the mechanism of switch on and switch off can be taken care wisely in an operon region. Now, all these three regions are clearly present in E. coli, and the absorption of glucose or the use of glucose is based on three different regions, which are this: the repressor protein, promoter, RNA polymerase play a very vital role for it. I hope you are clear with this concept of what is an operon and what does operon consist of. Thank you.